Hello children, in today's video 4 of the same chapter, molecular basis of inheritance, I will be taking the topic protein synthesis. We are going to learn the components required for protein synthesis, and later in the next video on the very same topic, protein synthesis, we can have a visual effect on it to visualize the two phases of protein synthesis. In this video, we are going to deal with the components for the two phases of protein synthesis. So, let us get started. Before we could start with protein synthesis, let us go into the central dogma in molecular biology. This was proposed by Francis Crick. It states that the genetic information flows from the DNA to the mRNA and from the mRNA to the proteins. Once again, the central dogma in molecular biology that was proposed by Francis Crick. States that genetic information flows from the DNA to the mRNA and from the mRNA to the proteins. The first step or the first process is called transcription. And the second phase or the second step is called translation. So, once again, the process by which an mRNA is formed from the DNA is known as transcription, and the process by which proteins are derived from the mRNA is called translation. In eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. So, if I depict the cell, this is the plasma membrane, this is the nuclear membrane, this is the DNA. Now, the DNA, from the DNA, mRNA is formed. This process is called transcription. It occurs in the nucleus. Now, this mRNA comes out through the nuclear pores. There are a number of pores in the nuclear membrane. It comes out through the nuclear pores into the cytoplasm, and in the cytoplasm proteins are synthesized. So, transcription inside the nucleus and translation outside the nucleus, that is in the cytoplasm. That is in the case of eukaryotes, whereas in prokaryotes we know there is no well defined nucleus. So, both transcription and translation occurs in the cytoplasm. See, this is the DNA, then it forms the mRNA and the mRNA forms the proteins. All occurs in the cytoplasm. So, as I proceed from one slide to the other slide, I will be giving you the difference between prokaryotes and eukaryotes. Now, we saw the first difference is where transcription and translation is occurring. In eukaryotes, transcription is occurring in the nucleus and translation is occurring in the cytoplasm. Whereas in prokaryotes, transcription and translation both are occurring in the cytoplasm or the cytosol, because there is no distinction of the nucleus from the cytoplasm, or there is no such a well defined nucleus in the prokaryotes, as you very well know. Another difference here itself I can highlight is in eukaryotes both transcription and translation occurs as a separate process. See, in the nucleus, transcription occurs, that is mRNA is formed. It is a separate process. After this transcription is complete, it comes out through the nuclear pore into the cytoplasm. So, transcription and translation are separate processes, whereas in prokaryotes, transcription and translation can be coupled. The meaning of this is, this mRNA is formed by transcription. Even before the transcription gets completed, translation can get started. So we can say transcription and translation are coupled. Even before the transcription is completed, even before completion, translation can be started. So this I've highlighted two differences between eukaryotes and prokaryotes. Now from the molecular biology point of view, it is depicted in this form. That is, from the DNA an mRNA is derived and that process is called transcription, and from the mRNA the proteins are derived and that is called translation. This is from the point of view of molecular biology. But from the biotechnological point of view, this can be represented in another way. You should be familiar with that representation also, because the coming two chapters we are going to deal is with biotechnology. The same thing is depicted in another way. See, this is the DNA. This is one strand which is running in the 3', 5' direction. Then the next strand which is running in the 5', 3' direction. We know it is running in anti-parallelly. So, the two strands of DNA. Now, it is only a small segment of the DNA that is transcribed or that is coded into the mRNA. Only a small segment. For example, I take only this segment. Only it is this segment that is coded into the mRNA. So, I am going to draw only so small mRNA. It should be a wavy line. Not only that, it is not both the strands that are getting transcribed, only one of the strand. And that strand, remember, it is the strand that acts as a template for transcription is running in the 3', 5' direction. Later I will tell you why this strand is only going to be transcribed into the mRNA. Now I got the mRNA. This is representing the mRNA. Okay. Now the next is protein. So now what is depicted is transcription. Next, translation. Translation is depicted like this way. We know that the building block of proteins are the amino acids. So I can show with 2 or 3 or 4 amino acids joined by peptide bond. This is one amino acid, the second amino acid, third amino acid. So I assume the proteins I have taken has 3 amino acids joined by a peptide bond. This is the representation for proteins. So this is the way when we depict it for biotechnology, and this is also known as gene expression. So when we talk from biotechnological point of view, it is called gene expression, and when we talk from molecular point of view, it is called the central dogma in molecular biology. So once again, if I explain in terms of biotechnology, we see the entire DNA with both the strands full length. Okay. Then only a small segment, I very clearly said only a small segment of the DNA and not both the DNA strands, only one of the DNA strand is transcribed into mRNA. Which is that DNA strand that is transcribed into the mRNA? It's the strand which is running the 3', 5' direction that forms the mRNA. Then from the mRNA what is synthesized? The proteins. And here I have taken a proteins which is having 3 amino acids. So 3 amino acid A, B and C joined by peptide bonds. Clear? And also remember the polarity of the mRNA is 5', 3'. Later we will understand with respect to the orientation, this 3', 5' and why is the 3', 5' is only the template. The next slide will learn that. This slide is only introducing you to two terms, central dogma and gene expression. So once again, if I raise this slide and start from the beginning, central dogma was proposed by Francis Crick, which states that genetic information flows from the DNA to the mRNA and from the mRNA to the proteins, and the two steps are transcription and translation. So what is transcription? The process by which an mRNA is derived from the DNA is called transcription, and translation is the process by which proteins are formed from the mRNA. Then in terms of biotechnology, what we call it? Gene expression, and it is represented symbolically like this. See, this is indicating the first step. This is indicating transcription, and the second step is translation. So this is the way how it is indicated in biotechnology, and it is called gene expression. That is, we have the two DNA strands, one running in the 3' 5' direction, the other strand running in the 5' 3' direction in full stretch, only a small segment. So, you see a wavy line, a very short wavy line. You can't extend this wavy line like this way. It should not be as big or as lengthy as the DNA. It's very wrong. Okay, it should be only short, because only a small segment of the DNA, and that too only one strand is being transcribed. And last we have, from the transcribed mRNA we get the proteins, and the proteins are indicated with amino acid joined by peptide bond. So, this template or this slide introduce you to two terms, very important, strong terminology, central dogma, which is made reference in molecular biology, and gene expressions in biotechnology. Clear? The next slide we are going to deal with protein synthesis. As I told you, the first step in protein synthesis is transcription. So, here it is indicated horizontally and here it is indicated vertically. So, first we will deal with transcription, and as I told you, only what is required, the components that are required for transcription will be dealt in this video. The two important components that you have to study for transcription, or that is the prerequisites of transcription, is the transcriptional unit, and second is the enzyme called RNA polymerase. So, first let us see what is a transcriptional unit. We know the transcriptional unit is the DNA. So, I will just draw the original DNA, which has two strands helically coiled. So, one strand, let it run in the 3' 5' direction, and the second strand running in the 5' 3' direction. So, they are running anti-parallelly. This is the transcriptional unit. Now, I am going to define all the parts of this transcriptional unit. I told you the entire DNA is not going to be transcribed, only a small segment is going to be transcribed. For example, let us select this segment to be transcribed. So, we have to define the parts of that particular region. There is a gene called the promoter, and there is another gene called the terminator, and between the two genes, between the promoter gene and the terminator gene, there are the genes called structural genes. So, once again I have defined that particular portion that is going to be transcribed. There is a gene called the promoter, which I have indicated in green. So, from that you can get a clue, it is a start signal, and then with red I have indicated the terminator. So, these are two important genes. There is a promoter and terminator, and both promoter and terminator, in between the promoter and terminator, what is present? The structural genes. Now, it depends. In prokaryotes, the structural genes are many number, not one, there are many. I will just divide it. See, let this be one structural gene, second structural gene, third structural gene. Okay, so let me demark it. This is one gene, this is second gene, and this is third gene. So, in prokaryotes, the structural genes are more in number, more than one. So, we call it polycistronic. In eukaryotes, on the other hand, the structural gene is only single. There is only one structural gene between the promoter and the terminator. Okay, if I scribble over this, there is only one structural gene. So, in eukaryote, it is monocistronic. So, I brought a third difference here. That is, in prokaryotes, how many structural genes are found between the promoter and the terminator? More than one. And each structural gene is also known as cistron. So, we call it polycistronic in prokaryotes, and you can remember also P for prokaryotes, P for polycistronic. Whereas in eukaryotes, there is only one structural gene between the promoter and the terminator, and so it is called monocistronic. So, I brought the third difference, and all these differences are added to a next slide. Okay. So, we saw the DNA, and I located the promoter with green color indicating start and terminator with red color. So only this part is going to be transcribed into the mRNA. So only this part is going to be transcribed into mRNA. So the wavy line is only going to come from this region. Okay. Now, and I told you between the promoter and the terminator are the what genes? Structural genes. So from this diagram, what and all I have told you, I have introduced you to 3 genes. The promoter gene, which I have indicated in green, which is a start gene, and at the end I put a terminator gene, and between the promoter and terminator genes I have indicated the structural genes. And remember, in prokaryotes there are many structural genes or many cistrons, therefore they are called polycistronic, whereas in eukaryotes there is only one structural gene between the promoter and the terminator, and therefore it is called monocistronic. Now the very same diagram I am going to depict by unwinding the 2 strands of DNA, which is depicted here below. See, this is the promoter, this is the terminator, clear? Then there are the 2 strands. I unwound the DNA, there are 2 strands. This is one strand, and look very clearly, look at the polarity of the strand. It is running in the 3' 5' direction, and look at the second strand running in the 5' 3' direction. And look here, this top strand is called template strand and the down strand is called coding strand. Now why is the top strand called template strand? Now you are going to learn the reason why the strand running in the 3' 5' direction is called template strand, and the strand running in the 5' 3' direction is called coding strand. Now here only you are going to learn why this is the template and this is the coding strand. So to know this, we have to learn the next component called the RNA polymerase. From the very term itself you will understand RNA polymerase is an enzyme bringing about or catalyzing the reaction called polymerization, that is adding of nucleotides. RNA polymerase, and the specialty of this enzyme is that it works only in the 5' 3' direction. Very important, the RNA polymerase adds the nucleotide in which direction? Only in the 5' 3' direction. So which can be the template? If it is adding the nucleotides only in the 5' 3' direction, which strand should I select? Should I select the 3' 5' strand or the 5' 3' strand? We have to select the 3' 5' strand. So it works in the 5' 3' direction. It should be anti-parallel. So which strand is the template? 3' 5'. It cannot be same. 5' 3' it's working the 5' 3' direction. We cannot use this strand which is running the 5' 3' direction. They are always anti-parallel. So always you have to have that concept anti-parallel. So never two running parallelly. 5' 3' direction, it's not going to be parallel, it should be anti-parallel. This always functions in the 5' 3' direction. Therefore, which will be the strand it will select for adding the nucleotides? It will select which strand? The strand which is having the polarity 3' 5', not 5' 3'. Clear? That's why this strand which is indicated in blue in this diagram is called the template strand. And the second strand which is indicated in green is called the coding strand. Very strangely, it has nothing to do with coding, but it's given the name coding and it has a lot of significance. This coding strand is very significant because it's very important. From the coding strand you can understand where to locate the promoter and the terminator. Remember, the promoter is located at the 5' end of the coding strand. Look at the coding strand. This is the 5' end of the coding strand. So I locate the promoter here, and the terminator will be located at the 3' end of the coding strand. So the significance of the coding strand is to locate the promoter and the terminator. Where is the promoter located of the coding strand? At the 5' end, and terminator is located at the 3' end of the coding strand. If at all we switch over this, interchange this, if I interchange this and put this as terminator and I interchange this and put this as promoter, what will happen? We have to reverse the polarity of the strands. Then which will be the coding strand? This should be the coding strand, because look, at the 5' end is our promoter. and look at the 3' end is the terminator. So it is with respect to the coding strand that the promoter and the terminator are located. So that's all about the transcriptional unit. Once again, after erasing, I'll explain transcriptional unit. The transcriptional unit is the DNA. I've unbound the DNA and I'm showing you the 2 strands. I'm redrawing it here. This is one strand which is running in the 3', 5' direction, and this is the second strand which is running in the 5', 3' direction. Now I am going to first of all label the strands. This strand called 3', 5' strand is the template strand, and the strand running in the 5', 3' direction is called the coding strand. The reason why it is called the template strand is because of the enzyme RNA polymerase. RNA polymerase functions only in the 5', 3' direction. So on which strand will it do, does the polymerization? It will select the 3', 5' strand. So that strand gets its name template strand. And the other strand, don't say second strand or up strand, low strand, no. The other strand is called the coding strand. The importance of the coding strand is it enables us to locate 2 very important genes. And the 2 genes I'll give in green, indicating the promoter, and red, that is indicating the terminator. I'm going to locate these 2 genes. Now, the promoter is always located at the 5' end of the coding strand. So I must locate it here, 5' end. And the terminator is located at the 3' end of the coding strand. So I have located the promoter and the terminator. Promoter, terminator. And which is the other gene that is flanked between the promoter and the terminator? It's the structural genes. Clear? So first you must give the 2 strands. The strand with the polarity 3', 5' is called template. The reason is because RNA polymerase functions in the 5', 3' direction. So which will be the template? The 3', 5' strand. So the other strand is called the coding strand. So you have drawn template and coding. It can be even reverse. You can put the coding on top, template below. Now you are going to locate the promoter and the terminator. Remember where you are going to place the promoter. The promoter should be placed at the 5' end of the coding strand and terminator at the 3' end of the coding strand. And so in between these 2, promoter and terminator, lies our structural genes. You can even reverse the polarity. If you reverse the polarity, here it's 5', 3', and this is 3', 5'. What happens? This down strand becomes the template and top strand becomes the coding strand. Then is the location of the promoter and terminator right? No. We have to remove the promoter and the terminator because we have to place it with respect to the coding strand. So our coding strand is on top, no? So on the coding strand 5' end only I am going to place what? Our promoter, and at the 3' end only I am going to place our terminator. Okay, it is with respect to the coding strand. I hope you have understood the 4 components of the, look at the transcriptional unit, the 4 components, namely template strand, coding strand, promoter, terminator. These are the very important 4 components. Once all these are placed, what is present between the promoter and the terminator will be the structural genes. And remember, for prokaryotes, there will be a number of structural genes. I am dividing it into many segments. 1, 2, 3, 4. There are many structural genes, so it's polycystonic. Whereas in eukaryotes, there is only one single structural gene. One single structural gene, and then it is called, in that case it will be called monocystonic. So now this is showing monocystonic, only one structural gene between the promoter and terminator. But if it is prokaryotes, it will be divided into many cystrons. Clear? So this is the transcriptional unit. And I told you about RNA polymerase is the enzyme that brings about polymerization. Next component, RNA polymerization, as I told you, it functions only in the 5', 3' direction. So, which strand it decides to take? It decides to take the strand which is running in the 3 prime 5 prime direction. Okay. So, the strand on which it works will be the strand running in the 3 prime 5 prime direction, because RNA polymerase functions in the 5 prime 3 prime direction. The role of the RNA polymerase is to bring the nucleotides or base pairs complementary to the base pairs found on the strand 3 prime 5 prime strand, and now we know the name of the strand. It is called the template strand. Okay. So, now over this example itself I am going to introduce, or I am going to place all the bases. For example, I am going to give some at random some bases on coding strand, and once I give the bases on the coding strand, I must find the bases on the template strand. I cannot give that at random, and it should be complementary to the coding strand. For example, for E's I am going with similar A 3 times, T 3 times, G 3 times, and C. These are the bases on the coding strand. So, what will be bases on the template strand? It will be complementary to the coding strand. For A it will be T. So, T. And for T it will be A. Then for G it will be C. And for these C's it will be G. So, you understand the base pairs, complementary base pairing. Now, which strand is going to form the mRNA? It is the template strand. So, I am going to draw the mRNA that is formed. So big, equivalent or coinciding with the structural genes. Now, this is going to be complementary here. For T, now remember mRNA is synthesized, and we know the basic difference between DNA and RNA. Instead of thymine, it is uracil. So, that you should have in mind. So, for T, what is complementary to T? It is A. So, A. See from here. For T, it is A. Now, for A, what should be the complementary? It's an RNA. It is U. See, U. Then for C, it is G. And for the G's, it is C. Look at the bases on the mRNA. The bases are complementary to the bases of the template strand. Now, if you closely analyze these bases, see, it is A, U, G, C. It is similar to which strand of the DNA? Look at both the strands of the DNA. Look at the blue strand and the green strand. The blue strand is the template strand and the green strand is the coding strand, and the stop in question is the mRNA. I am asking you, to which strand is the mRNA similar? Is it similar to the template strand? No, it is complementary. But look, it is similar to the coding strand. See, look. If I cross check, see, A. Here U. Instead of U, it is T. Then G. Here G. C. So, another significance of coding strand is the sequence of the coding strand is very much similar to the sequence in the mRNA. But what? Instead of thymine, it is uracil. In the case of thymine, what is placed? It is uracil. So, if they give you the coding strand and ask you to find out the sequence in the mRNA, you can write exactly similar to the coding strand, except T is replaced by U. So, therefore, I want to emphasize that coding strand has two significance. The significance of coding strand is, one, you are placing the promoter and the terminator with respect to the coding strand. That is, at the 5 prime end you are placing the promoter, and at the 3 prime end you are placing the terminator of which strand? Of the coding strand. And the second significance of the coding strand is, the coding strand is very much similar to the mRNA, except that T is replaced by U, because the one is a DNA, the other one is an RNA. So, that you should bear in mind, the significance of coding strand. And which part of the DNA gets transcribed? It's only a small segment, that is the structural genes. that is located between the promoter and the terminator that gets transcribed into the mRNA. And not only that, it's not both the strands that are getting transcribed, it's which strand that is getting transcribed? It is the strand that is running in the 3', 5' direction. Or what's the name of the strand? That is the template strand. So that also you should bear in mind. So the requisites of transcription is complete. The two main components are the transcriptional unit and two, the RNA polymerase enzyme. And transcriptional unit, I told you, remember the two strands, one running the 3', 5' direction, the other strand running the 5', 3' direction, and the strand which is running the 3', 5' direction is known as the template strand, and the other strand is called the coding strand. Then we are going to place the two genes. Which are the two genes? Promoter and terminator. How we are going to place the promoter and terminator? With respect to the coding strand. The promoter is placed at the 5' end and the terminator is placed at the 3' end of the coding strand. Remember, the genes that are going to be transcribed is only that sequence between the promoter and the terminator, and that sequence is known as the structural genes. For prokaryotes, there are a number of structural genes, whereas for eukaryotes, there is only one structural gene located between the promoter and the terminator. So only that is going to form the mRNA, that is a wavy line. And remember the sequence, another importance of the coding strands, the sequence of the mRNA is exactly similar to the coding strand, except that T is replaced by U. Clear? Then I told you another important fact is about the RNA polymerase. It functions only in the 5', 3' direction. Hence, which is going to be the strand that serves as a template? It is the strand which is running in the 3', 5' direction that is serving as the template. Now in the video I will be again saying you another difference with respect to prokaryotes and eukaryotes with respect to this particular enzyme RNA polymerase. But with respect to the components of transcription, I just winding up with just three differences between the prokaryotes and eukaryotes, which we will be once again reiterating in the next slide. The differences so far dealt. First difference, transcription and translation occurs in the cytoplasm in prokaryotes. So in prokaryotes, both transcription and translation occurs in the cytoplasm because there is no well-defined nucleus. Whereas in eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. Second difference, transcription and translation are coupled, which means even before transcription gets over, translation starts in prokaryotes, whereas in eukaryotes, transcription and translation are two different processes. Third difference, the structural genes are polycistronic. I told you between the promoter and the terminator in prokaryotes there are a number of structural genes, so we call it polycistronic and it is continuous. Whereas in eukaryotes the structural gene is single, there is only a single structural gene between the promoter and the terminator and it is split. This you will understand later. When I take transcription in eukaryotes, you will understand the meaning of the word split. Clear? So with so much we have dealt the three differences I have highlighted and put in this template. We will be adding two more differences to this when there are totally 5 differences. I will be adding 2 more differences when I deal with the transcription and translation in detail, showing you visuals. Clear? Next phase we know is translation. Now to understand translation, what are the components that we have to be familiar with? First component you have to be familiar with is the translational unit. Remember, the translational unit will be the product obtained after transcription. We saw in transcription is DNA. What we got as a result of transcription, the wavy line is the mRNA. So this is going to be the translational unit. I am going to describe the parts of this translational unit. Translational unit, I drew an mRNA and remember the polarity of this mRNA, it is running in the 5', 3' direction and there are bases. We know there are a number of bases. Now I am giving some bases at random, A, C, C, A, U, G, C, 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 G, 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 A, U, A, G, C, 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 whatever, some bases at random. A translational unit is an mRNA that comprises of a number of bases, and these bases are read in triplets. Remember how we read these bases. A in triplets, C, CA, UGC, CCG, GGA, AAU, AGC. Okay, now I have given it at random. So, these triplets on the mRNA are called codons. So, the first part of the mRNA we are learning is codons. So, what are codons? Codons are three consecutive bases on the mRNA are called codons. So, we see bases are there in the mRNA and how it is read in triplets, and these triplets are called codons. Very important parts of the translational unit. I told you the number of codons. There should be a start codon. I am giving it in the form of block. There should be a start codon and also there should be a stop codon. Now, the start codon is always AUG. You have to by heart this AUG. So, which is the start codon? AUG. It should be by hearted. And stop codon will be any one of the three. The three are UAA, UGA, or UAG. These are the three stop codons. I will select any one of these. UAA. So, this is the start codon and this is the stop codon. Clear? What else is found between the start and stop codons? There are the other codons, and remember it should be in triplets. So, I am going to give, for ease I am going to give it in with the same alphabets. C, 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 G, 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 A, A, A. So, now it is easy for you to read because I have given in the same alphabets, but it need not be like this way. It is jumbled and shuffled, C, A, G, G, C, A, U, G and all those combination. Okay, for you to identify, you can see how many codons in between the start and stop codon. There is C, 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 G, 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 A, A. So, how many codons totally are there in this translational unit? Totally 1 start codon, 2, 3, 4, 5. Fifth one is the stop codon. Clear? So, I have indicated an mRNA, and do not forget the polarity running the 5 prime 3 prime direction. So, which is going to be translated? This region is going to be translated. So, this is called the translated region, which is I am indicating with an abbreviation TR. Now, the region before the start codon and the region after the stop codon are the UTRs. What is UTR? Untranslated regions. So, what are the untranslated regions? The region before the start codon at the 5 prime end and the regions after the stop codon at the 3 prime ends are called the UTR. So, what is the middle? It is called TR. TR means translated regions. So, I am not making it more confusing. I am just rewriting or redrawing, showing the parts once again. Once again, a translational unit. I am drawing a straight line. I am indicating the start codon AUG and any stop codon UAA, and this region before at the 5 prime end and that region is at the 3 prime end. This region before the start codon is called UTR, untranslated region, and this region after the stop codon is known as UTR. UTR stands for untranslated region, and which is going to be translated into proteins? This region, that is between the start and stop. This is only going to be translated. This is the translated region. I am going to fill with some codons. Any random. Now I am going to give random figures. C, G, C, A, U, G, C, G, C, remember it should be in triplets. So, we have one codon, second codon, third codon, fourth codon, fifth codon. Okay, it should be only in triplets. So, we have the first codon definitely should be only AUG, that is start codon. This is my second codon, third codon, fourth codon, fifth codon, and here the sixth codon is the stop codon. This is a translational unit. Here also there may be bases A, C, 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 G, 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 whatever, A, C, G, B, G, 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 whatever be the bases, but they are untranslated. Only the region that is going to be translated is only between the start and stop. And remember, the bases are read in triplets called codons. So, that is the transitional unit, that is the mRNA. Next, amino acid. Next important component is the amino acids. You all are familiar with the amino acids. There are nearly 20 amino acids. Of these 20 amino acids, nearly 6 amino acids in different lessons, across different lessons we have learned. So, let us recollect the 6 amino acids. In the very same lesson we have learnt about the 2 amino acids which imparts positive charge to the histones. Recollect that. Which are the 2 amino acids that gives a positive charge to the histones? Arginine and lysine are the 2 amino acids which we have learnt in DNA packing, which gives positive charge to the histones. Then again, in the previous chapter we have learned about PKU, that is a disorder where phenylalanine is converted into the amino acid tyrosine in normal persons, but in these persons this conversion does not, in PKU cases this conversion does not take place because that enzyme phenylalanine hydroxylase is absent. So, again here we are learning two more amino acids, namely phenylalanine and tyrosine. So, I am listing 1, 2, 3, 4. Again 2 more amino acids we have learnt with respect to sickle cell anemia, 5 and 6, that is in the 6th position of the beta globin chain, which is the amino acid that should actually bind, is glutamic acid or glutamin. But what happens? A wrong amino acid comes in that position, that is valine. So, by way of learning other chapters, we are familiar with 6 amino acids. Once again, arginine, lysine in context to histones, how they are imparting positive charge to histones. Phenylalanine and tyrosine with respect to PKU. In normal persons, in us and all, the phenylalanine is converted to tyrosine, whereas glutamin and valine we have learned with respect to sickle cell anemia. In the 6th position of the beta globin chain, glutamin or glutamic acid should be present, but which a wrong amino acid is substituted called valine. So, we are familiar with 6 amino acids. So, again one more amino acid we have to be familiar, which is very, very important, that is methionine. So, to your list totally you have to learn only 7 amino acids by heart. You need not learn the remaining 13 amino acids. But remember, there are 20 amino acids, of which 6 you all are familiar with, and the 7th one, methionine, is very important because I told you here AUG, the amino acid that has to come to AUG is meth or methionine. Okay. So, this is the first amino acid that forms a protein chain. I told you this is going to form a protein chain, and I have represented protein chain in the form of amino acids. See how many amino acids will be formed here? 1, 2, 3, 4, 5, 6, not 6. The stop codon will not have amino acids, there will be 5, 1, 2, 3, 4, 5. And the first amino acid is always meth. Remember, for any protein, the first amino acid is methionine. It combines with AUG. We will be learning that in translation. As of now, remember, the first amino acid in any protein is methionine. So, we are having 7 amino acids that you have to learn for the 12th standard NCERT. So, that is about the amino acids. Next, ribosomes. The ribosomes, you all are familiar, it has 2 components, a large subunit and a smaller subunit. The large subunit is 50S and the small subunit is 30S. Therefore, our ribosomes in eukaryote is the 80S ribosomes. Okay, so remember the ribosomal units having 50S and 30S. a larger subunit and a smaller subunit binds at the mRNA at the start codon. Okay, it always binds at which codon? At the start codon, and remember our start codon is AUG. I am highlighting the start codon AUG. It always comes and binds at the start codon of the mRNA to start off protein synthesis. It has two units as I told you, 50S, the larger ribosomal unit, and the smaller ribosomal unit called 50S. Clear? It is a site for protein synthesis, only forming a site for protein synthesis. That is with regard to the ribosomes. Next, tRNA. tRNA you will have to learn in detail the structure of the tRNA. So, I will be taking it in a separate slide. So, let us move on to the structure of the tRNA. The structure of the tRNA was proposed by Francis Crick. So, second time in this video we are coming across Francis Crick. First he proposed the molecular, the central dogma in the molecular biology, and second time we are learning in context to the structure of tRNA, which was proposed by Francis Crick. This is the actual structure, inverted L. So, the tRNA has an inverted L. This is the actual structure. And the secondary structure of the tRNA is this, the clove leaf. It looks like the clover leaf. You play cards and you see the clove leaves. Okay, like this way you see in the cards. Okay. Now I am going to describe the structure of the tRNA from the secondary structure. So this is only the secondary structure, but the actual structure is the inverted L, and this structure was proposed by Francis Crick. Now talking about the structure, two main important parts. There is an anticodon loop. Look, there is an anticodon loop. And this anticodon loop has three bases, and these three bases are complementary to the bases on the codon. I told you on the mRNA there are bases, and those three bases or triplet bases are called codons. If the bases are AAA, the tRNA which is having anticodon complementary to AAA, which is complementary to AAA, it is UUU, will bind here, and this complementary base pairing between the A's and the U's. Clear? Here in this case, the codon on the mRNA is GAG, so the anticodon will be CUC. See, in the figure given here, the anticodon is CUC, so it will come correctly to the codon which is having GAG. It will not come to this codon, it will not come to this codon, here and here. It will come correctly to what is complementary to its anticodon. To C what is complementary? G. To U what is complementary? A. And to C it is G. So, it will come and bind here. So, this tRNA came and correctly bound here, not anywhere else. Okay. So, it has one region called the anticodon loop, which has bases complementary to the bases of the codon on the mRNA. And opposite to this anticodon loop is the amino acid binding site. Just opposite is the amino acid binding site. Here is the amino acid that is bound. Clear? So once again, the structure and the function of the tRNA was proposed by Francis Crick. The actual structure is an inverted L. See, you can see this diagram representing an inverted L. And the secondary structure is the clover leaf structure or the clove leaf. Okay. And two important parts, there is an anticodon loop, and here it is CUC. It complementary base pairs with the codon having GAG. So, you understood the function of this anticodon loop. This anticodon loop comes correctly to the codon which is complementary to it and form base pairs. Opposite to this anticodon loop is the amino acid binding site. So, here is the amino acid binding site. Then another very important specialty of the tRNA is that they are specific to the amino acid. For each amino acid there is a specific tRNA. Clear? Now, that is with regard to the structure of the tRNA. Now with regard to the function of the tRNA. The tRNA has a dual function. First function, it reads the bases on the mRNA. So the tRNA is going to read the bases on this mRNA. Look, it's going to read GUC, CAG, GAG, CUA, UAG. So first it reads and then it binds to the appropriate amino acid. Here, for example, GAG. So which is the tRNA that is going to come here? The tRNA which is having CUC. It reads GAG and binds to the appropriate amino acid. Now, to refer to which amino acid is going to come, we have like the Clark's table, you have to refer the genetic code. The genetic code is there in your text, you have to correctly find out. See the genetic codon, that is a codon on the mRNA GAG. So you cross check, where is GAG? Which is the amino acid that is going to come. And then when you check, you find out which is the amino acid. For GAG, the amino acid is glutamic acid. So that tRNA binds to the amino acid. And the binding of the tRNA with the amino acid is known as charging of tRNA or amino acylation. It occurs in the presence of ATP. It is energy expensive process. So once again, the function of tRNA. First, it reads the codons on the mRNA and then binds to the specific. So first it reads the codons on the mRNA and then binds to the specific amino acid. This binding of the tRNA with the amino acid is known as charging of the tRNA or amino acylation. Clear? So this is the function of the tRNA. It's having a dual function. And in this context, it is known as the adapter molecule. It is not only rigid, doing only one function reading, it is adapting. It is doing another function, binding to the appropriate amino acid and bringing it to that particular site. Okay, there are more verbs to add on. Actually, when you see the video, you can see reading, binding, bringing it to the correct site, and then this complementary base pairing between the codon and anticodon. But here they have explained with respect to two functions. That is, first it reads the codon and accordingly bind to the appropriate amino acid. This binding of the tRNA with the amino acid is known as charging of the tRNA or amino acylation, which is an energy expensive process which occurs in the presence of ATP. I hope you have understood the structure as well as the function of tRNA. And also remember another thing, tRNA are specific for amino acids. Not the same tRNA brings different amino acids. For meth, there's one specific tRNA. For proline, there's a specific tRNA. For phenylalanine, there's a specific tRNA. For serine, there's a specific tRNA. For each and every amino acid, 20 different amino acids, there are 20 different tRNAs. It's not that tRNAs are doing, taking turns and bringing different amino acids. Yes, very specific. So for the start AUG, that tRNA is called initiator tRNA. We know the start codon is AUG, and I told you to by heart AUG. So for that tRNA, the tRNA, you can guess if it's AUG, what will be the anticodon UAC of that particular tRNA, and it brings the amino acid methionine. It brings the amino acid methionine, and the anticodon is UAC and the codon is AUG. Okay, so this is an initiator tRNA. Clear? So tRNA are specific. There is initiator tRNA having the anticodon UAC, but there is no tRNA for the stop codons. For all codons there are tRNAs, but for the stop codons, which are the stop codons UAG, UGA and UAA, there are no tRNAs. Now moving on to the last concept, that is the genetic code. As I told you, it's just like the Clark's table for reference. You have to identify which amino acid is going to come to the site. So what you have to do is check which is the codon and refer the table and find out which is the amino acid that has to come. Just for reference purpose, but we have to elicit the features of a genetic code, which I will be explaining. So this is a genetic code, it is a checkerboard. It is a checkerboard of codons. Don't get confused, it's a checkerboard of codons. So what are you referring to? Codons. And what are codons? Codons are triplet bases found on the mRNA. See, this is the mRNA. 3 consecutive bases forms the codons. For example, I am going to give AAA, CCC, GGG, UUU, UAG. Okay. Now these are the codons on the mRNA. Now I am going to make use of this checkerboard. Now look at the codon. The first codon is AAA. So I want to know which amino acid is going to come at AAA. So cross check AAA, where? Here AAA. So which is the amino acid? You like the Clark's table just for reference. So what is going to come here? Lysine. Which amino acid? Lysine. Next codon is CCC. So look, C is for CCC. Look for CCC. Proline. See, again CC, it's proline. So which amino acid is going to come here? Proline. Next, GGG. Glycine. So glycine is going to come here. Then UUU. Phenylalanine is going to come here. Now next one, UAG. Look, UAG is a stop codon. It does not have a tRNA. Okay, so I'll elicit the features of a genetic code. So now you understand what is it. This serves as a checkerboard for you to check. Checkerboard for you to check the genetic codon, the codon on the mRNA, and cross check which amino acid is going to come. Okay. This checkerboard was proposed by many scientists, put their heads together to form this. And finally, it is a physicist named George Gamow who gave this permutation combination of 4 raised to 3. 4 into 4 into 4 giving 64. He gathered this from different informations. There are 4 bases, 20 amino acids, and each codon comprises 3 bases. So with all these informations, he brought it together and brought this combination, permutation combination 4 raised to 3, that is 4 into 4 into 4, which is equal to 64. So now this is credited to George Gamow. Actually, he is a scientist in physics, but he brought this mathematical, logical and implemented here and brought this in the form, in an organized way, in a checkerboard form. Okay, we have come across checkerboards in genetics, working genetics problems, but it's only dealing with two options from top and side. But here, from the options are coming from top, side, left, right, etc. to form 3. Okay, we are filling 3 in each. So from top you are taking 1, from side you are taking left, and from right you are taking, you are filling 3 bases. Okay, so this combination or the checkerboard comprising 64 codons, the credit goes to George Gamow. Now I am going to explain the features of this genetic code. The first and foremost features of this genetic code is that there are 64 codons, as I told you, 4 raised to 3 giving you 64. Of these 64 codons, 61 codons codes for amino acids. Remaining 3 do not code for amino acids. These 3 that do not code for amino acids are called the stop codon. So you have 64 codons arranged clearly. Of these 64 codons, 61 codes for amino acids, only 3 do not code for amino acids. Those 3 are UAA, you have to learn by heart, UGA and UAG. Let's figure it out in this. See, UAA, UAG and UGA. So we got the first feature. Second feature, of the genetic code is the start codon is AUG. Which is the start? AUG. Look, I am figuring it out here. Start codon is AUG and it binds with which amino acid? It binds with the amino acid methionine. Clear? Now, third feature, look here. It's easy for you to remember because it's UUU and you're looking only at the first quadrat. You get the third, fourth and fifth feature. UUU codes for the amino acid phenylalanine. Right from bacteria to man. The most primitive and the most modern advanced organism, right from bacteria to man, UUU codes for phenylalanine. This proves that genetic code is universal. So, which property it is proving? The genetic codes are universal with the very example of UUU codes for phenylalanine. But there are some exceptions with respect to certain protozoans and certain bacteria and certain mitochondrial genome. Fourth feature, again I am deriving the fourth feature from the same very first quadrat, UUU. Look at UUU. UUU codes for phenylalanine. Does that UUU code for any other amino acid cross check? No. So, remember this feature, codons codes for only one amino acid. So, you got that feature and this feature is called specific. There is a name for this feature, it is called specific. So, look at the codon, UUU codes only for the amino acid phenylalanine. Does UUU code for any other amino acid? Look at the 20 amino acids scattered here. No, you can't see UUU anywhere else. So, you can see that codons codes only for one amino acid. But look the other way around. I look this way. Now, look the other way around. But look at phenylalanine. Phenylalanine is coded by UUU as well as UUC. Look, it is coded by two codons. So, the fifth feature is some, it's not all, some amino acids are coded by more than one codon. And the very same example you can give phenylalanine is coded by UUU as well as UUC. You can see then leucine also. It's coded by UUA, UUG. Look at leucine, it's coded by 4 codons. CUU, CUC, CUA, CUG. Many, almost all amino acids are coded by more than one codon. This feature is known as degenerate. So, you got your fifth feature and the last feature. The last feature I cannot depict on this template, I have to draw an mRNA. So, I will just draw an mRNA on top with another color that stands out red. See, I am drawing an mRNA, I am giving the basis GGG, CCC, UUU, AAA, CCC, GGG. Okay. Now, how should this mRNA be read? Should it be read with pauses, punctuations or pauses? No, it should be read in a continuous manner. But at ease, because I am emphasizing the concept of codon, I took it in triplets, same letters for ease for you to identify that's a codon, but never, it should not be read with breaks, it should not be given because it is jumbled, it is at random, and also it should not be read with pauses or punctuation, it should be read at a stretch. So, what I dealt early, pausing and writing or giving the same alphabets is wrong. Never it occurs in our gene at ease for us to read GGG, CCC, UUU, AAA, wrong. It should be read in a continuous manner, GGG, CCC, UUU, AAA, CCC, GGG. But now because it's the same alphabet, you feel like it's like you can break it into fragments. No, it is not like it should be read in a continuous manner. What I am trying to say is that a genetic code is not punctuated or paused, it should be read in a continuous manner. So, once again the 6 features, the first feature very well you can elicit from the checkerboard. there are 64 codons, of which 61 codes for amino acid, 3 do not code for amino acid, which are the 3 UAA, UGA, UAG. You can figure it out here. Then, if you talked about the stop codon, the second feature you can talk about the start codon. The start codon is AUG and it codes for the amino acid methionine. So, first feature was the stop codon, second feature was the start codon. Third feature from the first quadrant. First quadrant you get the third, fourth feature, which is the third, fourth and fifth feature. The third feature UUU. UUU codes for the amino acid phenylalanine, and right from bacteria to man UUU codes for phenylalanine. This indicates that genetic code is universal. Again with the same example you can explain the next feature, specific. Look at the codon UUU, it codes for phenylalanine. Does this UUU code for serine? Does it code for proline? Does it code for leucine? Does it code for lysine? No, it only codes for phenylalanine. So, this property by which the codon codes only for one amino acid, it is known as specific. But on the other hand, did you see phenylalanine? It is coded by UUU and it is coded by UUC. So, we can say that amino acids can be coded by more than one codon. That feature is known as degenerate. And lastly, the codes are not read with punctuation or pauses, it is read in a continuous manner. So, these are the 5 important features of a genetic code, which is very, very important, and you must give it with examples. Especially the third, fourth and fifth, give with the example of UUU. It is very easy. UUU codes for phenylalanine. Here you have to use active voice and passive voice. When you are using codon with amino acid, you are using active voice. That is, codon is codes for one amino acid. But when you talk with respect to amino acid, you give it in the passive voice. The amino acid is coded by so and so codon. Clear? Remember, active, passive voice, make use of that. And also to make it easy, I can say a child can have one mother, that is only codon has only one mother, but a mother can have many children, that is only degenerate. You can also relate like that. Okay, but I feel to get the correct sequence or the frame of sentence, go with the active and passive voice. Here active, that is codon codes for one amino acid. Here the amino acids are coded by more than one amino acid, more than one codon. Clear? So, the 6 features that we can elicit from the checkerboard, and I told you who is behind making this checkerboard in this form. George Gamow, he is a scientist in physics who put forward this combination permutation of 4 raised to 3, 4 into 4 into 4 with the information of 4 bases, each codon having 3 bases and totally 20 amino acids, having therefore only 64 codons. I hope genetic code is clear. So, with that, what is required for translation is complete. I told you what are the important components, the translational unit or the mRNA. Then the amino acids, I told you by now you have to be familiar with 7 amino acids. In this topic we are learning methionine, which codes for the codon AUG. Then the ribosomes, which are the sites for protein synthesis, having the large ribosomal unit and the small ribosomal unit. And then I told you about the tRNA, it is cloud leaf in secondary structure, but in real structure it is an inverted L. And lastly, the genetic code, the 6 features of a genetic code, and George Gamow's contribution to making it into a checkerboard. I hope this is clear, and this should be very much clear to learn the next topic, which is protein synthesis. We are going to learn with the help of 3D animation video, so you will have a visual idea. So, when it is visualized, the concept will be connected, it will be conveyed clearly to you. Thank you.